I would like to acknowledge the people that really have done, the work, and basically here transmitting some of the work. And even though the title says just to talk more about the life cycle, I'm just going to go the different areas of what we miss or what we really don't know about Xyleborus glabratus. As you know, Xyleborus glabratus is originated, or the places where it has been collected has been from India, Myanmar, Taiwan, Bangladesh, Japan, and perhaps other countries in Asia, and several hosts are listed, Lydsea, Shorea robusta, Lauracea, and Fagaceae. However, our studies for Xyleborus glabratus have been really minimal in the area of origin. A very short survey in Taiwan where we placed logs of several of the species that I mentioned before were negative, capturing any Xyleborus glabratus. However, after looking at one of the host plants in a monosmophyllum in a collection, we finally found Xyleborus glabratus. But in the collection, the only time that we found glabratus was when glabratus was associated with termites that had been damaging the trunk, or with longhorn beetles that they were damaging the upper part of the tree. So from that point, even though there were really short associations, we really don't know much about the role of Xyleborus glabratus in the area of origin. Other work that we have been doing relates to avocado cultivar. Both Mayfield tested West Indies, Guatemalan and Mexican races of avocado. And since we have so many avocado cultivars in Florida, around 20-something, what we did was to place them in a no-choice experiment in contact with Xyleborus glabratus. And then we measured the number of holes per stem. And then with Jason Smith's lab, measure the frequency of lauricula. The interesting thing is the beetle bore into all of them, but the interesting part is lauricula didn't appear in three of the cultivars. Again, the trees that were used were small. I think this needs to be done again to verify why lauricula did not appear in some of those cultivars. So there are some indications, but again, some of these studies need to be done again. Other study that we did, and this is in cooperation here with California, was to expose logs of different species of Persea to Xyleborus glabratus in areas where Xyleborus glabratus was found, as well as placing them in the lab in contact with Xyleborus glabratus. Well, I was very happy during the first trials because there was at least one species that Xyleborus glabratus didn't bore into it. Then we asked Marilu Arpaia to send us some more fresh logs, and well, but the possibility of Flocosa not being a host for glabratus was something false that we were following after. So in conclusion, there is a lot that we need to learn about Xyleborus glabratus and the Persea species here in the tropics. This study started, still is, study in which we were collecting trees, exposing trees that they are common in the Florida hammocks and also in the swampy areas. And the goal for this experiment was to see that there was any repellency. My colleague Lucas Stelinsky also was sampling the same trees and seeing if volatiles from those trees would act as repellents from Xyleborus glabratus. To our surprise, bald cypress, Taxodium distinctus, and Annona glabra, that is a common tree in swamps of the Everglades, we obtained boring of Xyleborus glabratus. However, the lauricula was not positive for two of those. We did not inspect that the galleries was what we call successful galleries or not, but I think the good point here is that if these two were attractive or the beetle bore into that, it might be that some of these trees have some volatiles that can be used even for trapping, and that's what Stelinsky's lab is doing right now, trying to detect which volatiles are present in these ones and if they can enhance the current trapping situation for glabratus. With the behavior, we learned something really interesting. What we learned after observing the number of beetles that they were trapped. They were trapped from 7 a.m. until 7 p.m. in the evening, is that the beetle has what we call unimodal flight behavior, and this was the first test. And the second test also shows that during the dusk or close to the dusk areas is when you start collecting this large amount of beetles. And the correlation coefficient was negative regarding solar radiation. So when the solar radiation is becoming less and less, then is when you will trap more Xyleborus glabratus. What's the practical use? The practical use will be then for chemical control if applied at that time, when there are going to be more beetles around, and also for the growers when they are going to do pruning. If a grower is going to do pruning, it's advised that they will do it in the earlier hours in the morning where less Xyleborus glabratus are present. The seasonality, Jim Hanula did seasonality. This seasonality was done in Red Bay and Swamp Bay in Florida. And what it shows basically 6 to 7 generations per year. The lower peaks here, I think they are related that the hammocks were exhausted of beetles population, but you still can see some peaks in there. So basically, to me, it will be around 6 to 7 generations per year again in those hosts. But I would say that February to March will be perhaps the time where the highest peaks and perhaps when dispersal is occurring at the maximum time from one host to another during the year. So those are the practical implications of that. The life cycle opened a can of worms, really. We tested studied life cycle in avocado, Red Bay, and Swamp Bay. And the reason that we started testing this basically is basic knowledge that we needed, but also because in the collections of Red Bay, Swamp Bay, and avocado, we always observed that from avocado, very few Xyleborus glabratus were emerging. At the beginning, I thought that was competition with other beetles. Therefore, logs of these species were exposed to 20 females of Xyleborus glabratus. Water was provided, and the logs were split daily to study the development of this insect. Pardon me. This is kind of really interesting, and I have to come closer here. If you see here is the egg deposition, and this is for Swamp Bay. Here you have Red Bay, and then you have avocado. You can see that in avocado, the lower egg deposition, and as well, if you start going to larval development, pupa, and also the adults that they are emerging from avocado, you can see that these are more or less than what emerge per bolt of avocado. So there is something with these two species that logs of avocado lack. So if we go now for the number of successful boring of the beetle into each of the species, we really don't have any differences in there. But when the number of eggs are deposited, you can see that it's similar between Red Bay and avocado, but Swamp Bay has approximately 3 times the numbers that you obtain from both avocado and Red Bay. And the percentage of adults that reach adulthood is only 12% in avocado, is 48% in Red Bay, and 90% in Swamp Bay. What's going on? There's a difference in all of those cultivars that we don't know what's causing that, but I would like to talk more about that. But in general, the eggs hatch from 7 to 9 days when the temperature is around 96 degrees centigrade, the larva between 6 to 10 days, and the teneral, then the pupa between 4 to 7 days, and then you obtain the teneral adult. And these are the different characteristics. The egg, of course, is ovoid, clear looking. The larva, characteristic of a Scolytinae, a larva C-shaped, legless, and the pupa X-rayed. 3 larval instars, which is the common denomination for this species. And we also started an experiment that has not concluded right now, but the interesting thing is that around 12 degrees centigrade, we did not obtain an egg hatching or egg development, and pupal development did not occur at 12 degrees centigrade, which calls my attention in there. But the fastest, again, temperatures higher than 36 degrees centigrade, we did not obtain pupal development, and the same went for egg hatching. So there is a a difference in here, and I was surprised with the data, because when we observed the populations of glabratus in Taiwan was in high mountains, what we call coffee growing area, between 18 to 22 degrees centigrade. So I assume that in those places, glabratus was going to show a really good development even at low temperatures, but that doesn't seem to be the same anyway. This experiment has not finished yet. The gallery pattern is very interesting. We have a primary tunnel that is regularly 9 millimeters in length, and then they start branching to secondary tunnels and tertiary tunnels. Most of the eggs are found in the communal area. Sometimes a single egg is deposited, sometimes in groups. So this is found between the secondary and the primary tunnel. In the tertiary tunnels, we did not find the larva, which called my attention what we found in here. And regularly, the gallery length sometimes can go to 20 or more centimeters, and double the time in the width of the gallery length. In other systems, it follows this same ramification more toward one area than to another, but continues showing the primary tunnels, secondary tunnels, and tertiary tunnels. The gallery pattern was studied in Swan Bay, not in avocado. I wonder if the gallery pattern in avocado is going to have the same length that what we found in this host. Fungi in the galleries is a work that is in progress. Harrington and others found in the Micangia, Lauricola, other species of Rhaphelea, Arce, Subalba, but we really don't know which are the fungi that are found in the galleries at the different stages of development of that gallery. And Jason Smith is working with my student trying to determine which fungi will be present in which gallery. So these are work that is in progress. For me, the advantage to know this will be what's the nutritional value of those fungi. If we're going to find in the galleries the same fungi, I imagine that the ones that we're finding Xyleborus, glabratus, Micangia, is something that we need to find, but I still think this will be very good for diet development and to see what's the nutritional quality of this. Avocado, to me, then, is still a poor host, and I question why is avocado a poor host? Could be some of the chemical compositions present in avocado that are not allowing the fungi to develop as well as might develop in the other hosts. I don't know if that's the case or not, but I think that there are some practical implications in there. Emergence starts happening around 60 days after the female star completes their development, and proceeds to happen with peaks between 120 and 150 days after invasion of the log, and continues to 240 days after. So I have a very long pattern of emergence that is found. This is what we found in Swan Bay. We haven't measured what's the pattern of emergence in avocado. Larve rearing will continue to be based in what is the normal situation. Just collect red bay or Swan Bay infested logs, and we place them in containers in which we put a beetle collecting partition baited with a the manuka bait, and some paper towels that they are wet to maintain a high production in the laboratory. Now, the isolated colony can be produced, but I won't advise to use avocado to maintain a colony, because it's a really poor quality host for Xyleborus glabratus. So meridic diets, we have started many, but that is, I call it my fault, because anytime that we start doing that, I pull out the person that is working in the meridic diets, and we never have been able to determine if the diets that we have used, in which we have used red bay or avocado sawdust applied to it, are successful or not. So far, with the latest diets that we are trying, we are finding Xyleborus boring. Still, until last week, no fungal growth in the galleries observed. and other ones we found boring egg deposition in larval present, in which I believe there is some presence of the fungi. So in conclusion, first of all, the surveys need to be started really and continue in the areas of origin to give us a more clear picture of what's the role of Xyleborus glabratus in the area of origin. The nutritional value of fungi in the galleries, I think, need to be determined. The meridic diets we need to develop for further studies. And again, the low production of adults in avocado, that could be the Achilles heel that we might be able to use to avoid a higher epidemic in the avocado industry in Florida. That's it. Thanks.
Just to follow on about your point about the low adult survival in avocado, presumably 12% is pretty optimal, and in real life it'd even be lower than 12%. Do you think Xyleborus glabratus populations could sustain itself without, in avocado alone, without these alternative hosts like red bay and swamp bay around? Like, for example, in California, where you don't have those hosts there, do you think it could sort of reach, you have a self-sustaining population just in the avocado?
Well, it is obvious that the infestations that we are having right now in the commercial area are coming from the natural hosts, regularly swamp bay, that is present close to the commercial area. And so that's what the higher populations really are. Now, regarding California, California bay laurel is a host for Xyleborus glabratus. When we expose logs of bay laurel to glabratus, they was born into it. Paul Kendra and Bud Mayfield has pursued those studies and have verified that it does very well on bay laurel. So even though you don't have Persea bourbonia or palustris here, you have other susceptible cultivars for the population will build up more. So there is a possibility. So don't discount that.
Jorge,
Yeah.
You mentioned that one thing I didn't understand from your statistic that you put beetles on a log and then 60 to 240 days during that period you would get new beetles emerging. Is that
between 60 to 240 days you get emergence or beetle emergence?
So that is all from, let's say, you put in one mother and then her offspring reproduces on that log again too?
Yeah, yeah.
Oh, okay. All right. No, I thought it was just one mother.
It was there. Yeah.
This is a follow-up question to people who know the botany here in California. I'm wondering about the bay laurel. Is it occurring in the areas around the avocado orchards?
Grows everywhere.
Anybody?
Actually, yes. It's, especially in the coastal area of Santa Barbara, there is a lot of bay laurel in all the canyons. And it's right next to avocados in many places.
Okay.
One other question?
What is the explanation for the poor performance of the beetle on avocado? Is it related to the poor performance of the fungus, or the poor quality of the feed of the fungus as a feed?
Well, again, when we collected trees that have been affected by laurel wilt through Florida. From those trees, the whole trees were collected, put in cans, and we waited for emergence of Xyleborus glabratus. In all the cases, in some other places in Florida except Homestead, very small population of beetles emerged from avocado. These were from the trees that were infected. Again, from that time, my belief was that there was competition with other scolytids. But now, we exposed a single bolt. Of course, it's a dead bolt, and the tree might have deteriorated. I believe that there is something in avocado that avocado lacks or have that is not helping the growth of the fungus, but I leave that to the pathologist to find if there is something or biochemical, or is the substrate or nitrogen, if the fungus is doing well in nitrogen. I don't know if the amounts of nitrogen in the stem of avocado are less than in the other hosts. That I don't know. I think that's an area for more studies.
Thanks, Jorge.
Okay.